Hey everyone, and welcome back to STEM Powered. For today's activity, we're going to extract the DNA from a banana. Before we get to our experiment for today, let's review some important facts about DNA. DNA, or deoxyribonucleic acid, contains the instructions that tell our bodies how to grow and function. All living things from plants to animals have DNA. DNA has a double helix structure with alternating sugar and phosphate groups on its sides, and it's made up of 4 nucleobases, A, G, C, and T, that match up in different orders to code genes. A gene is the section of DNA that contains the instructions for how to make proteins. Alleles are variations of genes that are responsible for different traits. For example, the gene for brown hair is different from the gene for blonde hair. For this experiment, it's important that we know a few key things about the structure of cells. DNA is located in the nucleus, which is surrounded by lipids. Lipids are fatty substances that do not dissolve in water. A membrane of lipids not only exists around the nucleus, but it also surrounds the cell. Fun fact, did you know that bananas and humans share 50% of their DNA? To extract the banana's DNA, we are going to need 1/2 peeled ripe banana, 1/2 cup of hot water, 1 teaspoon of salt, 1/2 teaspoon of liquid dishwashing soap, 1 resealable quart-sized zip-top bag, rubbing alcohol, also known as isopropyl alcohol, a coffee filter or paper towel, a funnel, a glass, and a stir. Before you start your experiment, place the rubbing alcohol in the freezer so that it is cold by the time you use it. First, mash the banana in the bag for about a minute. Next, dissolve the teaspoon of salt in the hot water. Once it is dissolved, carefully add the mixture to the bag. Gently mix the salt water with the smashed banana, but watch out, the bag still might be hot. Then, add 1/2 the teaspoon of dish soap to the bag. Be gentle when you mix the soap with the rest of the mixture. You don't want it to become too bubbly. Next, set up your funnel and filter and slowly pour the contents of the bag into the funnel. It should take a few minutes for the mixture to filter through, but once it's done, add the rubbing alcohol that should have been cooling in the freezer this whole time. You should add about 1 to 2 inches of alcohol. After around 8 minutes, you should see the DNA start to precipitate, but the longer you wait, the more you should see. Once you are satisfied with the amount of DNA, you can take a wooden stir and move around the DNA to get a better look. I'm sure you're wondering, how do all of these somewhat random steps allow you to extract a banana's DNA? Well, when you mash the banana, you are breaking up the banana's cells and cell walls. This increases the DNA's surface area and allows more DNA to be extracted. The salt helps separate the DNA from the other materials. The positively charged salt ions surround the DNA's negatively charged phosphate groups and protect them from being dissolved in the water. Just like dish soap cuts through grease on your plates, it breaks apart the fatty lipids surrounding the nucleus. This releases the DNA. DNA is soluble in water, but not in alcohol. So when you add the alcohol, the DNA is pulled to the top of the mixture. Now that we know how the experiment works, let's check on the other club members' results. Good job, everyone. Those came out really great. If you enjoyed this experiment, you may be interested in a career with DNA. Some of the many DNA-related careers include forensic science, anthropology, genetics, biology, and archaeology. I hope you all enjoyed this video. Don't forget to like and subscribe so you never miss one of our activities. Bye!